We're going to talk about the life cycle of a cell. And I gave you a student version if you want it. At the end, we have a single slide recap that we will turn in. I do that with my first year bio kids, and I thought, maybe I should be doing that with you guys. So you'll have just a single slide to turn in for participation. Okay, so here we go. So when we're talking about the life cycle, right, we're talking about from beginning to end, obviously. The majority of the cell cycle is spent in interphase. We're going to need to do some math with that later this week. Calculating the percent time spent in interphase and the percent of time spent in mitosis. We say about 95% interphase and 5% mitosis, but different cells will have different amounts of time. So we're going to learn how to calculate that. Why would we even need to go through the cell cycle and produce more cells? It depends on the organism and it depends on the cell, right? So single-celled organisms obviously would go through a cell cycle so that they can replicate and keep the species continuing. For those of us that are multicellular, we need it in order to grow. We get bigger by adding cells. We also need to replace worn out cells and repair damaged cells. So that's why we would go through the cell division. Not all cells are dividing at the same rate. G0 phase might have been something you put on the new side of your notes on our old news, new news. Skin cells we lose every day, 12 to 24-hour cycle. So we're sloughing cells all the time, which is why we need to wash our clothes and our sheets on a frequent basis. Liver cells, however, they only divide 1 or every 2 years or when in stress. So these cells get arrested in this G0 phase where they're not preparing for division. Cells like nerve cells and red blood cells and muscle cells, they do not divide after they've reached maturity. They're going to stay in the G0 phase. So you're born with all the nerve cells you'll ever have. Red blood cells initially have a nucleus, but after they differentiate into a red blood cell, because they could become a white blood cell or a platelet as well, after they differentiate into a red cell, they lose their nucleus to make room for the hemoglobin. So it allows them to carry more oxygen. So if they don't have a nucleus, there's no way they could make more cells, right? So not all cells will follow the same cycle. Bacteria, for example, don't even replicate the same way our cells replicate. So bacteria, eubacteria or archaebacteria, and even amoebas, which are protozoans, they reproduce through a process of binary fission. So literally splitting in two. So with this, remember, bacteria have just a single circular DNA. Notice here, it attaches to the inner membrane, and then that one single circle will divide, make a copy of itself, I should say. So the DNA duplicates, it attaches both to the membrane, and then the cell walls begin to pinch inward. So they form a cell plate in between the two, and that's going to divide the cells. So that's as simple as it can be with binary fission and bacteria. So we'll watch this. Pretty simple, right? The cell's just dividing in half, which is not what's going to happen with eukaryotic cells. So here's a little bit more detail. So we want to look at how the bacteria's DNA is replicating so we can compare it later.
Most bacteria reproduce by a process called binary fission. During binary fission, the parental cell divides to form two daughter cells. In the first step of binary fission, the bacterium replicates or copies its chromosome and attaches each identical copy to a separate location on the cell membrane. Unlike eukaryotic cells, bacteria do not need to dissolve a nuclear membrane or assemble a mitotic spindle, making binary fission very quick and efficient. Second, the cell elongates. Since the chromosomes are attached to different locations on the cell membrane, they separate from one another. Third, the bacterium pinches off at the center and forms a septum, a separating wall between the two compartments of the cell. Finally, the cell splits into two new daughter cells. These daughter cells are fully mature bacteria that can grow and, if conditions are right, continue the binary fission process.
Okay. So I'm going to spend more time on the things that I think are new to you guys or less familiar to you. So I think binary fission, though we might have mentioned it, we didn't really talk about how it happened. So that's a good summary, I think, of the process. So eukaryotic cells are going to divide a little bit differently because we're more complex cells. We have way more genetic material, lots of chromosomes. We have organelles that have to duplicate themselves before they can divide. So it's a more complex process. We have three main stages. You will recognize interphase, mitosis, and cytokinesis. Interphase, remember the G1, S, and G2, gap 1, gap 2, and synthesis phase. So basically what's happening during interphase is the cell is growing. The genetic material is duplicating and the cell is preparing for division. Mitosis, notice much less of the time is spent in mitosis. That's the division of the nucleus itself. And then cytokinesis is division of the cytoplasm. So that's when we break the cell into two. All right, so we'll go into more detail on the mitosis in another day. Interphase is not a resting phase. A lot of people think that it's resting because it's in between phases, but it's really in between divisions, and you can't divide unless you have enough material to support two cells, right? So at this time, the genetic material is very thin and thread-like. You can barely see it. It's called chromatin. As you'll recall, we typically talk about chromosomes and chromatids when we're in mitosis. So G1 refers to gap, period of cell growth before the DNA is replicated. So basically the cell is getting bigger, adding cytoplasm. S phase, synthesis, is referring to synthesizing DNA, making more copies of each of the chromosomes. And then G2, you're preparing for division, so you're creating the proteins that are needed in order to move the chromosomes around and in order to divide the cell itself. So what this topic today is about is the control of those phases. What allows us to go from one phase to another, or what stops us from going from one phase to another. So we refer to the checkpoints. So we have stop and go signals, and they're molecular molecules. So we would say they're chemical signals that occur at critical points in the cell. So here you can see there's one towards the end of G1, there's one at the end of G2, and then there's one in the M phase. So these are our three key checkpoints. It indicates if all the processes have happened that need to happen in order for the cell to proceed and be a viable cell. Okay, so G1, G2, and M are your three checkpoints. You can see what's happening here at each of the checkpoints. So G1 is seeing if the cell is big enough. During G phase, we made copies of all our organelles as well as added cytoplasm to our cell. So is it big enough to become two cells? Is the environment favorable surrounding it? During the G2 phase, you can see over here, during G2, or S phase, remember, we're going to make more DNA. So at G2, did we make a copy of every chromosome? Is the environment favorable? Is the cell big enough? So we're adding on the DNA replication. During the M phase, this is metaphase, not mitosis, but metaphase. If you remember metaphase, when the chromosomes met at the middle, so are all of the chromosomes paired up and attached to the spindle? This would have to happen in order for each cell to get an exact copy of the genetic code, right? So those are our three important checkpoints. So what controls whether or not we can move to the next phase is a complex called a CDK cyclin complex. So cyclin gets its name because it cycles. So the amount of the molecule cycles throughout the cell. So you'll see that it's high during the point when it is ready to divide or ready to go to the next phase. CDK stands for cyclin dependent kinase. ASE endings tell us it's an enzyme, right? All kinases have to do with phosphorylation. So it will phosphorylate another molecule in order to either activate it or inactivate it. So this acts like an on-off switch. So the two will come together. There will be a phosphorylation, and that will allow us either to turn on or turn off the next phase of the cell cycle. So I have this video, I haven't added it in yet. I was trying to find an animation that would help you see that a little better. So this is what I have. It's not the one I want. And he talks really slow. So it's like you're trying to encourage him to keep going. But what I want you to notice is how it works. So this was the best I could find.
The group of proteins known as cyclins are an important set of master switches for various stages of the cell's cycle. These cyclins help to signal multiple proteins within the cell as to which actions are appropriate at a given stage of the cell cycle. There are different cyclins for different stages of the cell cycle, and production of new cyclins help to promote migration through the cell cycle as cells divide. Cyclin activity is mediated through a set of cyclin-dependent kinases, or CDK enzymes, which are in turn regulated by their phosphorylation. So, for example, in the cycle, a cyclin molecule will bind to the corresponding CDK enzyme. This changes its shape and allows for the phosphorylation of two amino acids, threonine 14 and tyrosine 15. This will inactivate the complex so that it can exist in the cell at a stage where it is not actively promoting cell division. Another position, the threonine at site 161, can later be phosphorylated as the cell has been triggered to divide. And then the phosphates on the threonine and tyrosine at sites 14 and 15 can then be removed. This complex will now phosphorylate target effector molecules, which will then migrate the cell through the next stage of cell division. A cell must be able to inactivate these complexes, allowing the cell to migrate to the following stage of the cell cycle. A small protein known as ubiquitin binds to cyclin and targets it for destruction. It will dissociate from the CDK and move to the proteasome in the cytoplasm, which is a complex which destroys targeted proteins. Once threonine 161 is dephosphorylated, the CDK has returned to its original starting state and the cycle can begin again. The group.
Okay. So you got the general idea that phosphorylating is going to be involved. I liked that it talked about how it could inactivate or it could activate in order to move you into the next phase of the cell cycle. I could have stopped at the end of that before they got into ubiquinone and proteasome, but both those terms are going to come up in the click and learn you're going to do today, so I thought I would leave it in. And when we get into chapter 15, we get into gene regulation. And we will find ubiquinone and proteasomes will be part of our gene regulation in turning off genes so that we're not making material when we don't need it, basically. So I thought I'd leave it in for that introduction. Okay, so you can see they come together and they make what's called a MPF, maturation promoting factor. So here you can see the levels of the cyclin cycle, and you saw in the video that it will be broken down, degraded after it's used, right? So we saw that in the ubiquinone tagging. So you see that cycling there. So this is your regulatory protein. And then CDKs will bind with them in order to activate them. So there'll be the transfer of phosphates in order to put it in an active form, as you saw in that video. And so that would be considered the MPF complex, which triggers us into the next phase. So here you can see those 3 checkpoints all have individual CDKs, not the same CDK. CDK cyclin complex, MPF. Okay, so they cycle, they bring us from one phase to the other. The proper regulation of cell cycle is so key to life, I'm going to read this exactly, that the genes for these regulatory proteins have been highly conserved. What do we mean when we say highly conserved? We mean throughout the kingdoms, this gene is fairly identical genetically in every single organism. So things that are absolutely essential for life, the gene isn't going to change very much because it has to work the way it has to work, right? So this is kind of important in allowing us, when we get later on into like genetic and technology. So being able to take genes from one organism and put them in a different organism and have that gene expressed, right? We need conserved regulation. That gene has to be the same in order for it to be able to transfer through the kingdoms. So basically the same in yeast, insects, plants, and animals, including us. Okay. So we also have, in addition to the CDKs, we have external cues. So you've probably heard of the growth hormone, for example, right? That's going to signal a cell to divide. So we have coordination throughout the body through our protein signals, and you can refer to them as hormones more specifically. We have density dependent inhibition, and anchorage dependent. So on the surface of the cell, they have receptors that work as touch receptors, touch sensors. So if they're butted up next to another cell, they don't need to divide, right? But if there is space beyond them, they will divide. So that's what that's referring to. Once it's too dense, or once it's reached its maximum density, it's not going to continue dividing. And the same thing with anchorage, there should be a basement membrane that they are attached to, and they won't go below that basement. So that is like touch sensors that regulate that. Interesting. Those of you who know Tigger from Miss Stabler's room, do you remember, or did you see him last year? He grew a second tail. Did anybody see that? Yeah. Why? Because somebody practically cut his tail off, and then we taped it up and we tried to like get it to fuse together and fix, but there was a gap in the tape, and so there was no anchorage for it. So it kept dividing to fill that gap because its touch receptors didn't tell it to stop because there wasn't anything outside of that. So it grew a second tail outside of that space. That's how that happened. And I just find that really interesting when I talk about this page. So this is just showing you the growth factor a little bit more. This would be like a human growth hormone, for example. So receptors, we talk about protein receptors on the surface of the cell membrane. And when it divides, it's going to cause a whole cascade of events inside of the cell that takes information to the nucleus and tells it to divide. Okay, so what happens if it ignores these signals? It ignores the anchorage dependence, the density dependence, the CDK complexes. Well, you're familiar with cancer, right? So we'll watch this short little TED video, I believe.
We all all start life as one single cell. Then that cell divides and we're 2 cells, then 4, then 8. Cells form tissues, tissues form organs, organs form us. These cell divisions, by which we go from a single cell to 100 trillion cells, are called growth. And growth seems like a simple thing because when we think of it, we typically think of someone getting taller, or later in life, wider. But to cells, growth isn't simple. Cell division is an intricate chemical dance that's part individual, part community-driven. And in a neighborhood of 100 trillion cells, sometimes things go wrong. Maybe an individual cell's set of instructions, or DNA, gets a typo, what we call a mutation. Most of the time, the cell senses mistakes and shuts itself down, or the system detects a troublemaker and eliminates it. But enough mutations can bypass these fail-safes, driving the cell to divide recklessly. That one rogue cell becomes 2, then 4, then 8. At every stage, the incorrect instructions are passed along to the cell's offspring. Weeks, months, or years after that one rogue cell transformed, you might see your doctor about a lump in your breast. Difficulty going to the bathroom could reveal a problem in your intestine, prostate, or bladder. Or a routine blood test might count too many white cells or elevated liver enzymes. Your doctor delivers the bad news. It's cancer. From here, your strategy will depend on where the cancer is and how far it's progressed. If the tumor is slow growing and in one place, surgery might be all you need, if anything. If the tumor is fast growing or invading nearby tissue, your doctor might recommend radiation or surgery followed by radiation. If the cancer has spread or if it's inherently everywhere like a leukemia, your doctor will most likely recommend chemotherapy or a combination of radiation and chemo. Radiation and most forms of chemo work by physically shredding the cell's DNA or disrupting the copying machinery. But neither radiation nor chemotherapeutic drugs target only cancer cells. Radiation hits whatever you point it at, and your bloodstream carries chemotherapeutics all over your body. So what happens when different cells get hit? Let's look at a healthy liver cell, a healthy hair cell, and a cancerous cell. The healthy liver cell divides only when it's stressed. The healthy hair cell divides frequently, and the cancer cell divides even more frequently and recklessly. When you take a chemotherapeutic drug, it will hit all of these cells. And remember that the drugs work typically by disrupting cell division, so every time a cell divides, it opens itself up to attack. And that means that the more frequently a cell divides, the more likely the drug is to kill it. So remember that hair cell? It divides frequently and isn't a threat. And there are other frequently dividing cells in your body like skin cells, gut cells, and blood cells. So the list of unpleasant side effects of cancer treatment parallels these tissue types: hair loss, skin rashes, nausea, vomiting, fatigue, weight loss, and pain. That makes sense because these are the cells that get hit the hardest. So in the end, it's all about growth. Cancer hijacks cells' natural division machinery and forces them to put the pedal to the metal, growing rapidly and recklessly. But using chemotherapeutic drugs, we take advantage of that aggressiveness, and we turn cancer's main strength into a weakness.
I liked how it explained how the more often a cell divides, the more it's affected by chemotherapy. And so that really helps us understand the side effects better and why like people lose their hair when they have chemotherapy. So I think that that was interesting. So basically cancer is a failure to pay attention to those different external cues and internal cues for division. So unrestrained, uncontrolled cell growth, meaning it's ignoring the checkpoints. So P53, that's what you guys are going to look at after the lecture. The P53 gene plays a role in the G1 checkpoint, the restriction point, and that it should halt division if there's damaged DNA. So it should identify that. So it will do one of a number of things. It might stimulate enzymes that repair the mutations and fix the DNA. It might send the cell out of the active dividing phase and into the G0 resting phase. It might keep a cell in G1 and not allow it to move on, or it might cause apoptosis. Remember, that's programmed cell death to get rid of the damaged cells. So these are all things that P53 should do. cancers have something wrong with your P53 gene. So if that P53 gene isn't functioning, it's not going to do any of these quality controls, right? So that's what you're going to investigate today after the lecture. This is showing you how P53 should work. This gene should identify damage to the DNA. Like right here, there's damage due to heat, radiation, different chemicals. And it should repair that division or that damage. And so that cell can continue on to divide, and the others will go through apoptosis. So if there's abnormal P53s, if there's a mutation in the P53 gene, or the P53 gene itself is very delicate, if you will, and it can be damaged very easily. So if it's damaged, it won't be able to do its job. So then when it divides and there's damage to the DNA of the cell itself, there's no control telling it to stop dividing. And so it continues to divide, and it creates that cancer cell that doesn't pay attention to any of these external cues, right? So there's unlimited growth. So it just keeps dividing. It ignores the checkpoints. They escape apoptosis, so it actually turns off the apoptosis gene so that doesn't occur. The checkpoints turn off the P53 gene. Let's see. Immortality, if you've heard of Henrietta Lacks, they've done some videos about her. She was a Black woman back in I want to say 50s, 40s, I don't remember exactly, but she had cervical cancer, and they took her cells without her knowledge, really, during a routine check. And they found that the cells would continue to divide forever in the lab, and they haven't had that ability. They had not had that ability for cells to divide continuously before. So her cells have continued to divide since then till now, and they're used in all kinds of labs for research. So there's the immortal thread of Henrietta Lacks, that her cells continue to divide. They promote blood vessel growth, so they turn on the genes related to blood vessels, and they overcome those anchorage and density dependent touch receptors. So you can see where you create a tumor rather than, a thin layer. They go beyond the anchorage, it get into the bloodstream, and they can metastasize, meaning that they move to other parts of your body. So you have a slide. If you could take a couple minutes, you have this one slide. I'm going to have you fill out these questions, and then we're going to go over the answers together, and then I'll be ready to turn you loose. So take a couple minutes. I'm going to be calling on people, and if you've already thought about it before I call on you, you'll feel better about answering. How is binary fission different than mitosis?
So like binary fission just basically like copies the DNA and splits, but mitosis has like a lot of steps to prepare for a viable daughter cell.
Okay. What are the three main phases of the cell cycle?
Interphase, mitosis, and cytokinesis.
Correct. What are the four phases of interphase?
The four phases of interphase are G1, S, G2, and M.
Mm-hmm. And M is actually not part of interphase. So there was a new one I introduced today, G0.
Oh, okay.
Yeah, so there's G1, G0, S, and G2. Function of the CDK complex.
To form MPF complexes and promote movement into the next phase of the cell cycle.
Okay. And what are some possible causes of cancer?
I said just when cells duplicate or reproduce too fast.
Okay. And how about Anatoly, some other possibilities?
I said if the P53 gene is abnormal, and then it can cause like uncontrolled cell division, or it can stop the checkpoints.
Okay. And Corbin, do you have any others that you listed as possible causes?
Apoptosis doesn't occur, so the cell keeps multiplying even after it's damaged.
Okay. All right. So you guys have that done and you're ready to turn that in. So you can go ahead and turn that into the Google Classroom. You will have in the Google Classroom, so you're moving into this click and learn. So I'm not sure if we've done a click and learn before. So the link is there, but this is the link also. So basically it's like an interactive PowerPoint. So along the way, you might have an animation or a video to watch. So here we just have things to read. Nothing's going to happen there. You'll click the arrows to move you around. Look for these sorts of things where they've added in an animation or a video, because you're going to get extra information from those. So there's only, I don't know, maybe 8, it says it right there, 8 slides. So it's not very long. This won't take you a whole lot of time to complete. But this is going to explore the P53 gene a little bit in a little bit greater detail. Ubiquitin and proteasome, you saw that in the video that we watched. So they show up in there as well. Okay, we've done this PowerPoint and we've done the recap. You're going to do the click and learn, and a couple of you are going to stick around for me to launch your test. Do you have any questions about anything else? I will see you Wednesday in the Zoom time. Other than that, do you have questions on anything else?